I hope you all had a good lunch. Welcome back. We're going to start this afternoon session with Curtin Marxer, who's a research assistant at the Davies Center at the University of Applied Sciences at the Grison, specializing in artificial intelligence and data analysis. He has a master's degree in science in business administration and information and data management. And he's also worked on the Liechtenstein Olympic Committee. Just fun fact, if you like. He's going to talk to us this afternoon on natural language processing as a way to get new drugs for repositioning glioblastoma therapy. And just for those of you who don't know, glioblastoma is an aggressive brain tumor, but I'll let him tell you more about it and not me. Welcome, Curtin.
Thank you. So, regardless of my previous background, I will not talk about drugs and doping today. So I'm more focused on, I'm glad that I can present our research on our case study on natural language processing NLP with open data for drug repositioning on glioblastoma therapy. Let's see if it works. Doesn't work. Beginner's luck. Thank you. First, I will give you a short introduction into the whole scope of research and the subjects, and also some background about the research itself. Then I will give you some insight in the methodology we used, and also present the results of discussion. And in the final steps, we will discuss some limitations and our plans for further research in this topic. First, about me, most was already told. I have a bachelor's degree in information science. I have a master's degree in business administration, information and data management, and I'm currently a research assistant at the Davies Center, the Center for Data Analytics, Visualization and Simulation at the University of Applied Science of the Grisons in Cour. For the background of our research, it was part of a collaborative project between the Davies Center and two neurosurgeons of the Cantonal Hospital of Winterthur. Our two collaborative partners developed a unique combined regimen of 9 repurposed non-oncological drugs with metronomic temozolomide for the treatment of glioblastoma. And it's called CSP version 9. And yeah, what I was already told, the glioblastoma is the most malignant brain tumor which you can get. Our overarching goal of the whole research project is to predict new possible drug candidates for repositioning by harvesting multiple data sources and using machine learning and deep learning techniques. This whole part of research was started as my master thesis with the main research question: how can unstructured data, text data, be used for drug repositioning, and how can their potential be used for to complement the databases? First, an introduction to the whole topic of drug development and drug repositioning. The development and discovery of new drugs is costly, risky, and takes a lot of time. We heard in the previous presentation in this track some challenges in the whole drug development process. And yeah, the success rate for a newly developed drug to reach clinically approved markets, worldwide markets, way below 10%. So only 10% of each drug development process gets finished. Many newly developed medical compounds end up abandoned because of these high failure rates. And yeah, the concept of drug repositioning and drug repurposing describes the process of identifying and developing new uses for already existing drugs and medical active compounds. We try to discover these new use cases by harvesting knowledge of previous research, of course, by using databases or text data. And it's highly beneficial for the commercial side of things and also for patients, especially for patients who have rare diseases where a development for a new drug is not that commercially attractive. Some strategies for drug repositioning. First, the most common is based on the idea that therapeutically observed side effects in clinical trials offer of drugs can also provide information about possible alternative uses. And we have the other approach, the similarity-based approach, where we try to find similarities of drugs or diseases, where drug similarity can be based on the combination of chemical properties, target structure, molecular target structure, or side effects. And the similarities in diseases can be concluded through ontologies or shared treatment profiles. What are the challenges with working with drug data? Drug data are highly complex and strongly interconnected. The amounts of biomedical data is steadily increasing, and also with that, number of available repositories and databases. And yeah, the data of these repositories and databases differ significantly in terms of scope, quality, and reliability, what causes some inconsistencies by choosing the right database. Yeah. So yeah, we have the challenge of selecting the right or the adequate database for our planned use of that data. As a short example, I will show you 3 different types of standardized databases for drug data for our case study glioblastoma. These are lists of drugs which are mainly indicated as drugs for the treatment of glioblastoma. On the left we see drugs.com, which provides 9 drugs. In the middle we have the main indications of drug bank, the most gold standard drug database, which has 4 main indications, but also provides 316 drug trials indications, which describes these are the drugs which are known to be in clinical trials. And then we have the drugcentra.org, which provides us with 7 drugs, but we can see the numbers differ, and also the definitions which drugs will be provided in these databases will differ. Interesting, on the left side on drugs.com we have the 2 separate entries of temodar and temozolomide, which are basically the same drug. Temodar is only the brand name of that drug. Yeah, what's the potential of text data? A huge amount of exclusive medical knowledge is buried in various types of unstructured text data, and also clinical reports and other medical publications. Especially clinical trials provide new knowledge that is often not yet recorded in the standardized databases and summarized medical literature, so on possible side effects or individual drug-drug interactions, for example. And the potential buried information is in the text data on probably abandoned medical compounds also. Yeah, what was the goal of this research first? First it was to test and evaluate different approaches or methods to predict new drug repositioning candidates using NLP, namely named entity recognition on open and publicly available data. And we try to determine the potential which that unstructured data has to combat found inconsistency in databases by filling those data gaps. For that, we developed 2 different workflows. One to identify concrete drug repositioning candidates for glioblastoma by combining different pre-trained biomedical NER and ER models, and we try to identify relationships between those biomedical entities. For that, we used SciSpaCy. It's a small biomedical NER model which provides fast and easy to use biomedical NER models. SciSpaCy is trained on the Genia corpus. And yeah, SciSpaCy also offers 4 different specialized biomedical NER models, which also enable a wide subject scope of applications with the provided labels of each individual model. What text data did we use? Based on the similarity approach, we extracted all clinical trials on the controlled vocabulary mesh term neuroectodermal tumors, which is a broader term for glioblastoma, which also includes, for example, medulloblastoma or optic cancer types, cancer types of optic cell of cancer cells. And we supplemented that data set with 3,259 95 abstracts of PubMed, and we had a resulting data set of 10,000 documents, which we analyzed. For that, we used with our first method classical co-occurrence analysis, where we try to find the most similar clinical trials by extracting only biomedical entities as relevant in that text data. And with those most similar document pairs, we try to extract and associate new drug-disease pairs. So if two clinical trials were pretty similar based on their occurring biomedical entities, we try to cross-associate them with each other. For that, we tested 4 different variations. One with the main model of SciSpaCy, which extracts every type of biomedical entity in those clinical trials. With our second method, we try to extract only genes, genomes, and gene products, and looking for similarities based only on that type. And with our third variation, we used diseases, symptoms, and side effects, which is often hard to do with labels because most symptoms are also labeled as diseases, and all diseases are could possibly be also symptoms. So we used the labels of our other model to reduce those other to like use a stopword list to reduce the results by using cancer and pathological formations, which got reduced from that data or from the results. With our last method, we try to use a more open approach by only tagging all cells, which could also be in that text data, and looking for the most similar document pairs. With our second method, we try to build chains of association by identifying new possible transitive relations to determine new repositioning candidates. We used the open discovery model of Swanson, which for that we extracted A-B relations from standardized databases, and then we used those B terms as search terms in our text data, so we could extract a new entity type by using NER from that text data. So we could associate new diseases, drug pairs. For that, we built 5 different types of those association chains. The first chain was a simple extracting disease gene associations from open targets, and where we use those entity types B as search terms. For our second method, we use gene variants. For our third, we use symptoms. And then with our fourth and 5 variation, we try to increase the length of that association chain by also by using 2 linked extractions from 2 different databases. First, we extracted the associated drugs from drug bank. Then we extracted the associated side effects from CIDR for each of these drugs. And then we used all those extracted entities as search term in our text data. Same for our last variations. There we extracted the cell lines from the genomics of drug sensitivity in cancer database. What type of extracted predicted candidates for reprocessing did we get? Basically, we first had chemical elements, which are also associated in most databases, for example, calcium or indium. We had chemical composite, of course, which is the most observed type, with, for example, O6-benzyl guanine. We had experimental vaccines, like example DNX2401, also called Tazadino-Turef. And we also found hormones and steroids, and also other types of drug reposition candidates like TT fields, which is an electronic impulsory device, which is also used for treatments. Now, how did we evaluate our candidates? Because we wanted to measure or to explore the potential of text data, we tried to benchmark all excited entities which what is known in the standardized databases, which is the state-of-the-art is DrugBank. So we all labeled our predicted categories into our candidates in 3 different categories. The first category was known in DrugBank. So all drugs which we found, which were also in DrugBank, were put in that category. Our second category was the potential unknown candidates, which weren't known to DrugBank. And then we had the invalid category, which was just bad tagger results or just general broad terms for drugs, for example, acid or the broader term which you can't really label as a drug or identify as a sole drug itself. Yeah, and then we we labeled these for both of our methods. Interestingly, the distribution of known to DrugBank was higher in our first method with 43% or 44%, and in our second we had a ratio of 39% for that ratio. And most interestingly, like both ratios of known and to DrugBank and potentially unknown candidates relate to each other, because the higher the overlap with DrugBank is, the higher could the predicted candidates of the potential unknown category candidates be an indication for their quality. So the higher the ratio of the known in DrugBank, the higher in quality probably the potential unknown categories are. These are just a summary of our results of our all employed variations, where we use the pairwise cousin distance between each clinical study of 0.2 for most specialized thing. What you see here is that the cell lines and component approach provide the most number of predicted drug candidates, but also showed what we also can see is that the gene and genomes variation showed the lowest ratio of potential unknown candidates and the highest overlap with DrugBank, which could indicate that searching for genes probably provides the best results. For our second method, in the first row we can see the number of extracted search terms which you got from the databases, where the side effects, of course, the gene variants provide the most terms, but at the same time predicted the lowest number of predicted drug candidates. Furthermore, we could see that by using the difference in, oh sorry, I lost the plot. Sorry, and also the disease drug cell line also provide a low number of predicted candidates. Some conclusions in general. The analysis of unstructured text data can enable a more comprehensive overview of potential drug candidates. There were case examples in our of our predicted drug candidates like Sixotumumab and paracetamol, which were identified as possible drug candidates which were not in DrugBank, but after concurring research, we found that they are currently in clinical trials. So get those results, and that overlapping genes as markers showed the most reliable candidates for repositioning through our methods and use case. And that also rarely any gene variants and rare lines were documented in our selected data in the clinical trials summaries, and also that only probably symptoms and side effects and genes seem suitable for those types of embeddings in future research by analyzing text data. What were the main limitations? The publicly available clinical text data are mostly only provided as short abstracts, which is not that ideal because most of biomedical NER models are trained on full text data. And yeah, there was a lack of objective qualitative assessment of our identified repositioning candidates yet. That's what we'll try to do in the next step. And yeah, we used only a vague association approach, so we didn't analyze each associations based on their exact semantic connections, except maybe in method 2 for the first extraction of the standardized association pairs. And yeah, we couldn't detect their possible causalities or positive and negative relationships. And yeah, general, the poor performance in the size-spacey NER models, which are worse than other available biomedical NER models from Stanza or from Spark NLP. Further research, yeah, we're gonna increase our data sets, of course, so we don't only use text data from the same scope like Glioblastoma. So we try to include a more heterogenic text data set on other diseases and to analyze those found drug-drug interactions in each study. And yeah, thank you very much for your time.
Thank you very much. We have time for a couple of quick questions. Yes, please, at the back.
Yeah, that's a good question, but the problem with using large language models on this topic is that you need biomedical trained models on that specific scope, and by using large language models, you can't reconstruct why those two drugs were associated with each other. That's why we used NER, because you can detect, okay, which two entities are the cause for those similarities. So we have more, you can go back and analyze why are they similar, which you probably can't do with large language models.
Thank you. Is that a question?
Yeah, that's the next step we will try to do. Because this was more my master's thesis, it started as a master's, we didn't include that much embeddings at all from the biomedical fields. But yeah, that's the most relevant question. Like, can we get more information on, for example, gene expression data on drugs in this whole process, and other available data sources to also to score each predicted drug repurposing candidate.
Is there a last question? Yes.
Sorry, I didn't fully understand. How did you exactly label those invalid results? Did you have a rule-based approach, like for generic terms?
Yeah, of course. Yeah, exactly. We labeled every bad tagger results by only numbers, which could also happen with the size-spacey model. General broad terms were also a rule, and yeah, we used a rule set for every candidate, which include there.
Okay, let's thank the speaker again.
Thank you very much.
Thank you. Thank you very much.
Thank you.